This picture shows different types of centrifuge machine. They are used to separate the content of a sample based on the density. You can set the speed or resolution per minute according to your requirement. Make sure to put the sample in a balanced form or even number to avoid imbalance during centrifugation. This is a picture of milli-Q water purification system. It is important in cell culture that the milli-Q water is being used in preparing sterile pure water for media preparation, phosphate buffer saline, cleaning the bottles, and also for preparation of RNase-free water for PCR work. There are three types of purified water. Type 1 is known as the ultra pure water. It's an ion-free with low total organic carbon content. The ultra pure water is recommended for sensitive laboratory application such as advanced analytical techniques, example HPLC, the high performance liquid chromatography, the ultra high performance liquid chromatography, the UHPLC, for liquid chromatography mass spectrometry, ICP MS or inductively coupled plasma mass spectrometry, and also for molecular biology applications such as PCR, sequencing, and to prepare buffers and culture media for mammalian cell culture. Type 2 pure water is recommended for general lab use, such as to prepare buffers, reagents, culture media, and as for feed for another equipment. Example would be type 1 system, the clinical analyzers, dishwashers, stability test chambers. So in this case, type 2 pure water can be used. Next is the type 3 pure water. The type 3 pure water is the lowest grade of pure water, and it is produced by reverse osmosis. The type 3 pure water is normally recommended for non-critical lab application such as glassware rinsing, heating bath, autoclave, and as feed for type 1 water system. This is a picture of an autoclave machine, or known as sterilizer. The autoclave machine is used to sterilize consumables such as pipette tips, glass bottles, glass beaker, fluid such as sterile water for media preparation, phosphate buffer saline solution preparation, and other materials or liquid that is required and used for cell culture work. Before you push the start button, you must ensure that the water level at the bottom of the autoclave machine is within the normal level. This is an oven, or known as a drying station. After the consumables have been sterilized, they need to be placed in an oven to dry up all the excess water accumulated during sterilization. Usually the temperature is set between 40 to 54 degrees Celsius. This is a picture of water bath. Water bath can be set at certain temperature to thaw frozen liquids such as media, FBS, penicillin, and streptomycin before use. Please ensure that the water bath is always kept clean prior use, as it can be one of the source of contamination in cell culture. This picture shows the cryo-freezing container. The cryo-freezing container is also known as Mr. Frosty to some people. In cell culture, storing the cells in liquid nitrogen is important in making sure that the cells are viable for a longer period of time. For storage in extreme coldness, cells needs to be stored in cryovials. Before that, The cells needs to be stored in different temperature and they are placed in a specialized container.